Logo for the National Center for Advancing Translational Sciences, Case Studies in Translational Science. 7 icons are arrayed at the bottom of the screen containing the following text: focus on unmet needs, boundary crossing partnerships, efficiency and speed, creativity and innovation, generalizable solutions, cross-disciplinary team science, and bold and rigorous approaches. Video title text: Developing Tissue Chips to Transform Drug Discovery and Development. Passley Hargrove-Grimes, Ph.D., NCATS Office of Special Initiatives.
I'm Dr. Passley Hargrove-Grimes from the NCATS Tissue Chip Program. At NCATS, our mission is to develop more treatments for more patients more rapidly. To achieve this, we're working on a tool that we believe will transform the drug discovery and development process. Every day, millions of people take over-the-counter medications and prescription drugs. But did you know that only 7% of human diseases have available and effective treatments?
Why is that?
There have been incredible advances in basic scientific knowledge and techniques over the past 2 decades. Yet most research to develop new medicine fails. Traditionally, scientists develop medicines for people by testing promising drug compounds in cells and then move on to research in animal models. But that approach doesn't consistently and accurately predict whether a medicine will work in people. This means that a great deal of time, money, and effort are often spent testing drugs that ultimately fail in humans. So what can scientists do to improve the chances that a promising drug will work for people?
Improving how medicines are developed.
At NCATS, we're building an innovative tool to help the scientific community increase its success rates when developing new medicines. This new tool is called a tissue chip.
Images of tissue chips. One is the size of a USB stick. The other is the size of a credit card. Both are made from clear plastic, and the channels and chambers that contain tissues and fluids are visible.
These tiny bioengineered devices are made of human cells. They're able to replicate key functions of human organs and tissues.
Tissue chips are not miniature human organs on chips.
Instead, they're smaller, functional pieces of human organs. We can use these devices to test potential new medicines and predict which ones will work in humans far more accurately than the approaches that we traditionally use. Tissue chips have 3 main features that separate them from other ways of testing potential medicines, such as using cells or animal models. First, tissue chips have a 3D tissue configuration, much like a normal human organ. Second, tissue chips are composed of multiple cell types. And third, tissue chips include biomechanical forces. That means, for instance, that they can mimic the stretch forces that lung tissues experience when you breathe in or out. These features make tissue chips a much more effective way to develop and test potential medicines. Tissue chips could dramatically improve the accuracy, speed, and cost of discovering and developing new medicines. Consider a recent forecast by experts from 15 different pharmaceutical companies. They predicted that within 5 years, tissue chips could save between 10 and 26% of lab research and development costs. Lowering the cost of developing a new drug is a key way that we can accelerate the delivery of more medicines to more people who need them. Tissue chips could also make it possible to develop new drugs without testing them in humans, preventing risk to human participants, including vulnerable patient populations, such as the children and elderly. Tissue chips would also make it far easier to test potential medicines for rare diseases, because in many rare diseases, there may not be enough people or patients with the condition to meet the requirements for a scientific study. All of these tissue chip benefits can help us accelerate research to develop more drugs and lower the risk for patients. And this would benefit patients with both common and rare conditions. Collaboration and partnerships are critical to tissue chip success making tissue chips a core part of our toolkit in developing new drugs. With funding from NCATS, scientists across the country have created tissue chips for nearly every major human organ and tissue system.
3D vector illustration of a human and the following organs: brain, ear, muscle, heart, lungs, kidneys, liver, intestine, reproductive systems, blood vessels, and skin. Other illustrations include fat and a disease model.
And these include but aren't limited to the brain, the heart, lung, liver, skeletal muscle, and female reproductive systems.
Just to name a few.
These tissue chips are now being used to conduct research on common and rare conditions, and they're even being used in studies of potential treatments for COVID-19. For example, researchers at the University of Washington have created a kidney tissue chip to study kidney diseases. One of the rare diseases they're researching, focal segmental glomerulosclerosis, has no current treatments, and very few clinical trials have been created for patients with this rare disease. Collaborators also include patient advocacy groups. They've provided blood and tissue samples from patients that can be used as a direct comparison to the tissues that are used in tissue chips. This direct comparison is essential to ensure the accuracy of using tissue chips to mimic functional aspects of the human body. The pharmaceutical industry has also partnered with the NIH Tissue Chip Program to write manuscripts describing how they would design and use organ-specific tissue chips for drug development research. In addition, federal agencies,
such as the Food and Drug Administration,
have offered regulatory guidance on how tissue chips can be certified as approved drug development tools. NCATS and other NIH institutes are funding research projects that use tissue chips as a tool to transform clinical trials. For example, rather than exposing patients themselves to risks in drug research, scientists are creating tissue chips from patients' own tissues to test potential drugs for safety and efficacy. We call these tissue chips patient surrogates because they can safely and effectively take the place of actual patients in a clinical trial.
3D illustration of human cells from an organ collecting onto a tissue chip.
Ultimately, these patient surrogate tissue chips can shorten the initial phases of clinical trials. They also help us better understand which patients are more likely to respond positively to drugs and medicines. Those patients whose tissue chips don't respond to a treatment can still sign up for other clinical trials where they may yet find effective treatments and cures for their disease or disorder. At NCATS, it is our goal to have researchers turn tissue chips into an indispensable tool for developing new medicines and therapies. Tissue chips have the ability to transform the drug development process, saving both time and money, increasing success rates, and delivering effective new medicines for a broad range of conditions and diseases. If you are a scientist, a patient, or a patient advocate who's interested in collaborating with NCATS on tissue chip technology, we would love to meet with you to learn more about your potential interests. You can learn more about this initiative and about ways you can get involved at our website. We look forward to collaborating with you.
Learn more about NCATS's tissue chip initiatives. URL ncats.nih.gov/tissue chips. NIH and National Center for Advancing Translational Sciences logo.